Good morning, everyone. Welcome to special session 1. Moving beyond home ranges. How can we harness fish movement ecology to study, understand, and manage ecosystem functioning? My name is Julian Lilkendey, but fortunately I can't be there with you guys today. Funding, am I right? Renato, Robert, and I are excited to extend a warm welcome to our diverse group of speakers, each bringing a unique perspective and expertise to the session. Their contributions are essential in weaving together a rich tapestry of knowledge that spans various aspects of fish movement ecology and ecosystem functioning. This session is intended to be more than just a series of talks. It's a collaborative effort. To integrate diverse research areas, we aim to create a dialogue that bridges the gap between fish movement ecology and the broader study of ecosystem functioning. This integration is crucial for developing effective conservation and restoration efforts, especially in a world where ecological and socio-economic systems are increasingly interconnected and vulnerable. Our speakers will share their latest research, shedding light on the intricate patterns of fish movements. These insights are not just academic pursuits. They are vital pieces in understanding the complex puzzle of ecosystem functioning. By tracking the transfer of energy and nutrients through aquatic systems, our speakers will help us grasp the broader implications of these movements. Their presentations will contribute to a deeper understanding of how these patterns affect and are affected by the dynamic changes occurring in the Indo-Pacific. Now, we have in store for you 5 talks focusing on tropical and subtropical ecosystems, 2 talks focusing on fishes and their functionality in temperate ecosystems, 3 presentations focusing on what I would call grand-scale redistributions, and finally 4 studies focusing on marine macrofauna. Renato, Robert and I look forward to the rich discussions and innovative ideas that will emerge from this session. Together, we hope to pave the way for new approaches in studying and managing the functioning of ecosystems, ensuring their health and resilience for future generations. Now, without further ado, let's start with the first presentation. My most recent work focuses on rapidly measuring in situ rates of energy expenditure in fish. In ecological research, in situ information on energy expenditure of free-roaming animals are vital for our understanding of how animals adapt their energy use strategies in response to environmental changes. This is particularly pertinent in degraded ecosystems, where altered resource availability can significantly impact foraging behaviors. However, accurately gauging metabolic traits of animals is challenging, especially in aquatic environments. By combining 3-dimensional high-resolution velocity data with allometric scaling of metabolic rates, we aim to shed light on the energetic underpinnings of species interactions and ecosystem trophodynamics. By comparing energy expenditure between species and between habitats of different ecological status, this approach allows us to assess ecosystem functioning with unprecedented precision. We used calibrated stereo video setups comprising a set of 2 GoPro cameras to capture video footage at a resolution of 60 frames per second. In an open source software called BitSync, we manually determine functional traits such as bite rates and distances between consecutive bites in brown surgeonfish and in the yellowtail tang. We also developed a pipeline for AI-driven fish detection, identification and tracking from our stereo video footage. We calibrated our system using MATLAB with a checkboard pattern and performed stereo rectification using OpenCV. For object detection, we employed the YOLO convolutional neural network, retrained with still background images from our videos. We trained our recognition algorithm with location-invariant fish species images from iNaturalist for more accurate species classification. We then integrated the deep sort framework for multi-object tracking, enabling reliable automatic detection and tracking. By employing median smoothing and Kalman filtering between detections, we were able to produce fish trajectories at a temporal resolution of 12 Hz. Our method involved frame-by-frame analysis of fish coordinates to calculate differences in velocity and acceleration. Recognizing that high-precision measurements of acceleration in 3 dimensions correlate with energy expenditure in free-roaming animals, we used this approach to estimate the energy expenditure of individual fish during foraging. This method is called overall dynamic body acceleration and sums up the absolute values of acceleration in all 3 axes, x, y, and z, capturing both acceleration and deceleration. We estimated the mass of individual fish by using automatically measured lengths and applying length-mass relationships sourced from FishBase. We then applied allometric scaling, which relates body mass to metabolic rates to enhance the accuracy of our estimated rates of energy expenditure. For this, we use standard metabolic rate mass relationships from surrogate species with similar functional roles, specifically Stenochatus striatus and Zebrasoma scopas, from Schittekate et al. 2022. Nina, great study by the way. The mean distances between consecutive bites were significantly greater for yellowtail tang compared to brown surgeonfish. The observed rates of energy expenditure, on the other hand, were significantly higher in brown surgeonfish when compared to yellowtail tang. Our functionally highly important model species exhibited a divergence in energy use strategies. Brown surgeonfish expended more energy per unit time while foraging, potentially due to its specialized feeding strategy. In contrast, yellowtail tang appears to have a more energy-efficient, generalist foraging approach, expending less energy per unit time. Expressions of metabolic traits and distinct optimal foraging strategies of herbivorous fish influence the health and resilience of coral reef ecosystems. The study of energy use strategies among fish species, as revealed by our innovative methodology, will be a valuable lens through which we can understand modifications to ecosystem functioning. This study was funded by the Fisheries Society of the British Isles. If you are interested in novel approaches to analyze continuous movement data, please check out Amagan Sabishian's talk this afternoon. I just can't get enough of that smile. I will be available on Mentimeter in the next hour to answer questions online. But now, I would like to give the floor to our talented master student Emily Yap. Have a great session, everyone.